Welcome, everyone. I'm JT Vannah, your host today from Neuroelectrics. Thanks for joining us for an illuminating panel discussion where we'll explore the dynamic intersection of brain health and neurotechnology. In an era where mental health and well-being is paramount, understanding how movement impacts brain function is crucial. Our expert panel will delve into cutting-edge insights on how neurotech can enhance mental health through personalized neuromodulation and innovative brain stimulation techniques. We'll discover unique details and practical applications that can revolutionize your approach to mental health and wellness. We're thrilled to have you along to learn about this pioneering research and connect with leaders in the field. This program is the third in our quarterly panel series in celebration of Neuroelectric's 10th year in Boston, Massachusetts, in which we focus on themes surrounding mental health challenges. Today, our topic is Brain Health Gets Moving with Neurotech, and this fall, we'll explore suicide prevention by adapting brain circuitry. We hope you'll join us for this important issue as well. So let's introduce our distinguished speakers for this event in the order that they will appear. First up, we have Dr. Mark Hartman. Dr. Hartman's research delves into the psychological and neurological aspects of physical activity, particularly focused on the neural underpinnings of exertional fatigue and the mechanisms behind physical activity and behavior and adherence. His current projects explore the use of transcranial direct current stimulation to modulate exertional fatigue and enhance exercise adherence, aiming to develop potential treatments for clinical fatigue. Next, we have Dr. Mark Muthalib, founder and lead researcher at SilverLine Research and research associate at the University of Montpellier. Dr. Muthalib has over 15 years of pioneering academic and research expertise in human neurophysiology and neuromodulation. Dr. Muthalib brings a wealth of knowledge to the table. His impressive career spans more than 2 decades as a health and research consultant, underpinned by a PhD in human neurophysiology. Mark's diverse qualifications in entrepreneurship, technology innovation management, and computer science further amplify his ability to drive innovation and bridge the gap between cutting-edge research and practical applications. Finally, Dr. Brendan McNeish, who is an assistant professor in physical medicine and rehabilitation and neurology at the University of Pittsburgh, a Pepper Scholar, and a dad of twin boys currently surviving the terrible twos. Brendan practices neuromuscular medicine at the University of Pittsburgh and specializes in the treatment and rehabilitation of neuromuscular conditions due to cancer and its treatments. Brendan's research interest is in understanding the contributions of peripheral nervous system and central nervous system to mobility in older cancer survivors to inform novel mobility interventions, including neuromodulation. Following the presentations, we will engage in a panel discussion where our speakers will provide insights and will address questions raised. Thanks for everyone being here today, both to our speakers and our viewers. So without further ado, Dr. Hartman, the floor is yours.
Okay, so thanks for this opportunity to talk about some of my research. I'm going to be discussing using non-invasive brain stimulation to increase exercise adherence and enjoyment. I am assistant professor at the Department of Kinesiology. Now, I study exercise, but one of the unfortunate aspects of exercise is that no one does it. So as an exercise psychologist, this is quite interesting to me. And if you study exercise and measure it objectively, only about 10% of the US population achieve the minimum guidelines for health of 150 minutes of MVPA per week. Now, if you get people to exercise, they sign up for a randomized controlled trial, they have access to trainers, a beautiful facility, they even get paid. After about 6 months, they drop out. So I'm quite interested in that. Now, if you ask people, why don't they exercise? Number one reason, unsurprisingly to many, is lack of time. But if you ask a second question, how many hours per day are you on social media, watching TV, and playing video games? The average American spends between 6.5 and 7 hours of leisure screen time per day, mostly occurring in the evening. So there is time there. So it's perhaps maybe not a lack of time, but a lack of prioritization of time. And if you look at reasons 2, 3, and 4 that people provide, these are psychological constructs that are highly related to behavior. So maybe there's something more telling about those answers than the lack of time. So one of the most fundamental principles in psychology is the pleasure behavioral loop. As humans, if an activity feels pleasant, we are more likely to do it again. If an activity feels unpleasant or evokes displeasure, we are less likely to do it again. So in our lab, we study affective responses to exercise. And affect is simply just a core basic feeling state that underlies all emotions and moods. And it can be positively valenced, neutral, or negative. And we measure affect in our lab using the feeling scale, just simple unipolar or bipolar scale that you ask a participant either every 1 to 5 minutes during an exercise session. They simply indicate their response, and we track that response over time. Now, some of the early studies to look at the association between how individuals feel during exercise and their subsequent exercise behavior found a pretty strong relationship. So individuals, if they scored an exercise session, let's say a +3, and you compared that to someone who said, I'm a +4, that's a 1-unit change. This study found that 1-unit change, that +4 person was exercising an additional 38 minutes more than the +3 person 6 months later, and 41 minutes more at 12-month follow-up. And that scaled. So if you compared a +3 person to a +5, that's 76 additional minutes of moderate activity at 6 months and 82 additional minutes at that 12 months follow-up. So we know that exercise and affect responses are dependent upon intensity. So this is a graph showing a graded exercise test on a bike where the pedal resistance is slowly increasing until the person maxes out. And early stages of the exercise, they'll feel pretty good, maybe a slight increase or plateau. And there's a threshold, a point at which they start to feel worse. And that's usually nonlinear, it's quadratic. They rapidly decline until they achieve exhaustion and quit, feeling the worst. Now, if you look at the brain, and specifically we looked at the dorsolateral prefrontal cortex, and I'll explain kind of why we looked at that reason in a moment, you see a similar pattern. So measuring oxygenation using something like NIRS, we can index the neural activity, and we see that early on in the exercise, you see this increase in oxygenation up to a point, up to a threshold, where all of a sudden there's this rapid nonlinear decline. And in fact, it goes well below baseline. This kind of extreme deoxygenation occurs at the point of exhaustion. And indeed, these two graphs do correlate. The drop in oxygenation in this area of the brain is associated with the drop of the feeling scale score, or feeling worse. So why the dorsolateral? Well, in the psychology and neuroscience literature, many studies have been done in fMRI scanners looking at what areas and what networks of the brain are activated during exercise. So this is one study analysis looking at several dozen studies, not in exercise context, but just looking at areas that were associated with emotional regulation. Many areas, of course, lit up, but the most common area that's involved in this response is the dorsolateral prefrontal cortex. So in the exercise context, the idea is how you regulate, or your ability to regulate how you feel during exercise, is what's helped you reframe the exercise stimulus as either something good or bad, and may actually help you push through an unpleasant feeling during the exercise, which is maybe why athletes are so good at this. So the question we looked at in our lab is, okay, can we improve affective responses to exercise by attenuating this decline in oxygenation of the dorsolateral prefrontal cortex if, in fact, these two things are functionally related? And so we used TRNS, which is transcranial random noise stimulation, which is kind of a newer stimulation technology. And the advantage of this is you can stimulate two areas of the brain simultaneously without having to worry about a cathode or reference electrode that could be potentially deactivating the area. So it's a alternating current at a random frequency. We use 60 to 500 hertz, and it works by a principle called stochastic resonance, where simply if you inject the optimal level of noise into a system, it will enhance the activity of that system. And previous studies using this technology in the brain has shown it to increase the probability of neural activation. So that was the stimulation technique we chose. And we had individuals exercise for 25 minutes on a bike, including a 5-minute warm-up. This was a single session online, so during the exercise. They came in twice, one with a stimulation on and one with off. And we simply measured how they felt every 5 minutes during the exercise. And we found that by minute 25, they did feel significantly better, those who received the stimulation, compared to those who did not. Now, of note, you can see kind of a divergence of this graph occurring between minute 15 and 20. And previous studies have shown that the effects of TDCS or TRNS usually takes about 10, 15 minutes to take effect. So we're looking at applying an offline stimulation perhaps for 10 minutes prior to the exercise, then continuing that stimulation during the exercise to see if we can see this convergence in how individuals feel earlier, and perhaps that can be magnified over time. Another application that we are currently using this for is with clinical population. specifically patients with chronic obstructive pulmonary disease, COPD. With these patients, exercise is the most beneficial treatment available. However, it's probably the one activity they want to do the least because of how uncomfortable it feels. They get dyspnea. It's a very unpleasant thing for them to do, but very effective. So can we use the stimulation on them to make the activity maybe more pleasant, or at the very least, less unpleasant, and thereby feeding into this positive cycle of physical activity by increasing their motivation to want to do it again? They're going to get more fit. That's going to help the breathlessness, et cetera, et cetera. So in this, it's a 2-arm study. So they only receive the stimulation or they do not. And instead of a single session, it's a multi-session, 8 weeks, twice a week for 20 minutes, looking at affective responses to exercise, overall enjoyment, and specifically looking at dyspnea ratings. Is it affecting the breathing, the perception of the breathing? And then looking at that mechanism of oxygenation in the dorsolateral prefrontal cortex. In patients with COPD, oxygenation is limited to other areas of the body, such as the exercising limbs. We want to see if it's being affected here in the brain. And then looking specifically, as this will be paired with a pulmonary rehabilitation program, are we changing their adherence? Are they attending more sessions? Are they less likely to drop out? Again, hopefully linked to the fact that they'll be feeling a lot better during that exercise. So that's the brief overview of my studies that I'm currently working on, and that concludes my presentation. So I am happy to answer any questions at this time.
Thanks, Mark. We will hold the questions till we get to this discussion portion. But thank you. That was a great presentation. Mark Muthalib, you're on deck next, whenever you're ready.
Okay, thank you very much for the invitation to present some of my research and some of the products that we're developing in this field of neurostimulation and neuroimaging. So I think Mark already indicated what near-infrared spectroscopy can be used for. Just wanted to show some background to what is actually measuring in comparison to EEG. Most people are more familiar with EEG. So we have the neurovascular coupling, blood flow increase, representing the increase in neural activity. The measurements of the NIRS response, this large increase in oxyhemoglobin, the blood flow increase, also a decrease in deoxy, is created by a mixture of neurometabolic coupling, neurovascular coupling, and blood flow responses. So at the end, what we expect from an increase in neural activity is a greater increase in oxyhemoglobin with a greater decrease in deoxy. That's kind of what we're normally looking for in this hemodynamic response, similar to fMRI. So how does it compare to the standard neuroimaging technology of fMRI? So we did this study in Kiel. So what we see here is that the nice correspondence for a motor task, finger sequence or finger tapping, on the motor cortex. So we measure the same area with EEG, fNIRS, and fMRI. And we see this nice increase and decrease of the hemodynamic response, which is represented in the BOL response in fMRI and the NIRS. So it's measuring the cortical activation in the simplest terms. So my foray into TDCS, I was working on electrical muscle stimulation at that time during my PhD in Australia in around 2006-07, and came across the use of TDCS for fatigue research. So we were looking at integrating our protocol of fatigue with measurements of NIRS. And as Mark pointed out nicely about why we measure from the prefrontal cortex, is explained in that sense. So we were interested to replicate a paper that was just published by one of the pioneers of TDCS in Italy, Professor Priori, who did an isometric contraction at 20% and until you have this fatigue point. So in this graph on the bottom right is the point of fatigue. And we wanted to see if TDCS applied after a first fatigue session, 1 hour after, would reduce the residual fatigue, as Koji Mani in the previous paper at that time published in 2007. It took a while for us to publish this, but essentially what we showed was that in young healthy males, we didn't see this prolongation or a reduction in the residual fatigue as Koji Mani, who utilize more older population. So there could be a factor of the fatigability. Now, I don't want to go into detail about that. So the idea is that once you're in a state of residual fatigue, then adding a TDCS in that state could be more beneficial than somebody who could recover within an hour. So in this case, we didn't show an effect of TDCS, but the important aspect was that the NIRS as Mark indicated, at the point of fatigue was quite consistent. So it was a good marker of when somebody would achieve a level of fatigue. So from this work, the work I mainly did came over to Montpellier in France to develop a fully integrated system and to explore how well NIRS and EEG could be utilized to understand better the TDCS effects. So we, after some development from around 2012, we had the first project where we did an online, so during a task, a motor task, so finger sequence, and we did TDCS at the same time for 20 minutes. So we wanted to see if during the task, if TDCS is implemented compared to when the TDCS is placed after, the motor task. So what we found here is that when you normally do a finger sequence task, you find an increased activation. So this is 16 channels, 8 channels in one hemisphere, 8 channels on the other. So the right graph here would show, let's say, the circular areas would be the motor cortex region, and we have a larger increase for right finger sequence task on the left hemisphere, and vice versa for the opposite hand. So what we found was that when you did online TDCS, the level of activation was reduced. So since we didn't change the performance of the task, we have to assume that it's a change in efficiency of the brain to perform the task. In this case, since we didn't do separate online and offline stimulation conditions, we weren't sure if the offline effects were affected by the online. So here we had my PhD student completing this work where we looked at 3 different conditions: online, offline condition, and sham condition. And essentially what we found was this red circle would represent the online condition, similar to the previous study. We found a reduced activation, indicating a greater efficiency during the task performance, and an offline, actually an increase. And the sham, compared to sham, that didn't show much change in the NIRS response. However, about half an hour after the task performance and stimulation, we found a reversed effect. So whether this could represent plasticity occurring is what we were trying to understand of the process of consolidation. In this case, it was purely 30 minutes after. We have done some other projects where we actually did TDCS over 3 days to look at whether online or offline or a preconditioning protocol of TDCS has better learning capacity. But in this case, we were trying to see how well NIRS can be used to indicate the level of immediate plasticity changes. And in this case, we showed this like a biphasic response of a reduced activation during the task and an increase in activation half an hour after, which could be the starting point of plasticity occurring. So the clinical aspects of my research extended to patient populations in Parkinson's and stroke patients mainly. And so we looked at how best to provide an integrative atmosphere for the patients to perform exercise or movement in this case. So in the top left would be a Parkinson's patients performing a VR game where they have to move while they're getting stimulated. So these are projects that have been ongoing. While the project On the right is just using NIRS while you're doing a virtual reality game. So the main project that we've been working on here in Montpellier, which is an extension of the motor task that we've been working towards, is using TDCS, a high definition montage, during VR training in stroke patients. So these patients have paresis in the hemisphere, from the hemisphere opposite to the lesion. So if the left hemisphere is more damaged, with the right hand is more affected. And here we're trying to recover this arm function by stimulating and measuring the responses using NIRS and EEG. So the top figure here, graph will show the layout of the locations for the NIRS channels, and then the NIRS and EEG channels on the right side. And the bottom panel shows NIRS, EEG, and the high definition TDCS electrodes in one cap while the patient is doing the VR game. So this is all wireless communication, frees us from having the wires connected most of the time. Just some preliminary results of comparison between stroke patients and healthy elderly. So this is the system where online collecting of the NIRS and EEG at the same time, using LSL, as well as the kinematics, so movement patterns. So what we see is in stroke patients, just doing a pace reaching task when they're given directions of each reaching and back movement, patients are doing it slower and with more trunk movement. So at the same time, the EEG is indicating event-related desynchronization, which is a level of, say, excitability of the motor cortex, is lower in stroke patients, while the NIRS response is higher. So this would show there's an inefficiency in the patient population compared to healthy elderly to do the same task. It's more analysis and more tasks we do as a circular task. I won't go into that. But essentially, this is showing how well the two NIRS and EEG can be used with kinematic data to provide a full picture of how comparison population, then we'll be able to see if patients' recovery correspond with normalizing of these responses. So another aspect of what SilverLine does is, with StarStem and the NIRS system, is that we provide training to set it up, as well as providing software, OptiSim software, that can provide real-time analysis of the data. So if you're running a study, you'll be able to see in real-time analysis, so you don't have to wait till post-processing. So it's a quick test bed in some sense. If you do have this equipment, you can already start using this software. So even if you just have a StarStem with the EEG, we have a lot of parameters that can be online assessed. But of course, you can also do stimulation with that. We have some programs to do closed loop stimulation, which measures the NIRS or EEG response and then stimulates accordingly. And we'll be able to provide training on how to optimize this as part of our support service. Another aspect of how we're developing products, maybe for mental health, we see that there's an issue of stress in general with the population, and we're looking at solutions for this. And we've developed a single channel. We're working with our partners who've developed very nice compact NIRS system, that one channel system, very lightweight. And we developed an app that can interact with the subject or the patient, and they can start doing breathwork training, for example. But you can also use the same system for, let's say, for exercise. You can wear this and run an exercise session and already get quickly your NIRS changes. So it becomes very practical when you're at home-based monitoring, or getting somebody to do this outside of a lab or a clinic. So we haven't got this system out yet, but we will be able to roll this out in the next few months. Okay, so I think, thank you very much.
Thank you, Mark. Very, very interesting. Brendan, you're all set whenever you're ready.
All righty. So again, I'm Brendan McNeish. I'm a physician scientist at University of Pittsburgh in the Department of Physical Medicine and Rehabilitation and Neurology. The title of my short presentation here is Applying TDCS for Multimodal Rehabilitation in Older Cancer Survivors, a High-Need Population. And so I want to introduce this population because this might be a new population that you might think about neuromodulation in. And so cancer survivors, they're a very growing population thanks to better diagnosis, specifically earlier diagnosis, and better treatment. And there really is a rapid increase in cancer survivors. And that rapid increase is occurring in older adults. And you see here the statistic that 67% of all survivors are 65 or older. And within that breakdown, you can see this is a graph with years. On the x-axis, Y is the prevalence, and each color is a demographic of age. And if you just look at it, it's really striking that, purple and green are rapidly increasing. And these correspond to older adults between 65 and 84. And we know that in the aging population that they have really a lot of issues with mobility. And so cancer has new types of risk factors, including cancer, as well as its treatment that might change some of these impairments that these older adults may get. And so what does cancer survivorship look like for older adults? Well, the physical function of this population is significantly impaired compared to their cancer-free peers. They're at a higher risk of falls. They're at a higher risk of balance impairments. They have an increased trajectory of declines in gait speed and increased fractures and loss of independence. And so these are, really strong indicators that the physical function is declining. And it's even more rapid than their cancer-free peers. So, why might this be occurring? And the first player on the block has been this condition called CIPN, and CIPN is chemotherapy-induced peripheral neuropathy. And so chemotherapy can be neurotoxic to your peripheral nerves, and this can reduce, say, proprioception or distal weakness. And this might be related to impairments. And so this has been shown to be associated with increased falls, 2.5 times, increased fractures, likely from the falls that are occurring, and along with decreased balance. And the adage in the older cancer survivor literature for people who've been exposed to chemotherapy is that it is your peripheral neuropathy that is causing your functional mobility deficits. However, you see a nerve here, and the type of neuropathy that occurs is an axonal neuropathy. And when you lose your axons in your peripheral nervous system, there really is limited recovery. They grow back at, basically a millimeter a day, a centimeter a week, an inch a month type of deal. And so there really isn't good recovery. So it's probably not surprising that rehabilitation interventions targeting neuropathy have really not improved mobility in this population. And so the question would be, what could be targetable? And the question is, alcohol causes damage to your peripheral nervous system, but it also damages your central nervous system. Diabetes is another neurotoxic mechanism, and it damages your peripheral nervous system, and it also damages your central nervous system. And chemotherapy, while it's neurotoxic to your peripheral nervous system, also damages your central nervous system. And so I think it's not. So my next question that I want to try to see is the brain associated to mobility in cancer survivors, specifically those that were chemotherapy treated? And so this is a recent pilot study that we did, and we really just looked at executive function neuropsychological tests and how that was related cross-sectionally to people's balance, standing on one leg. And you see here reaction accuracy. This was done by a go, no-go task. And you can see as they are better at inhibiting and better at doing the go, no-go tasks, their balance was also better. And then trails a set shifting executive function task. You see as that goes down, their performance is worse. Their standing on one leg was also worse. And so what we kind of concluded is that executive function capacities were associated with balance. And then what I don't show you here is that they were really strongly associated to recurrent falls in any falls. And all these findings were independent of the neuropathy itself. And so this was kind of the preliminary data for my career development award that I submitted and is actually being reviewed today on study section. So if I'm a little anxious, that's the reason why. Or maybe one of the reasons why. And really, my question is the brain a mobility neurosubstrate? And potentially, could this be a way to improve mobility in older cancer survivors that are treated with chemotherapy? And so you can see some of the, I think probably the most important aim in this, well, one of the important aims is trying to maybe quantify the peripheral nervous system here on the right, along with the central nervous system and its contributions to gait during a dual task paradigm. It sounds like there's some interest in NIRS here, and I'm trying to enter that as well. And so PFC activity, that's going to be measured by NIRS. And so those are the first two aims, kind of. And then the last aim is really assessing the impact of a one-time session of TDCS in these older taxane-treated breast cancer survivors in their performance during the dual task gait protocol, as well as how it changes the PFC activity, the cortical hemodynamic response. And so that's really how I'm starting to use TDCS as kind of my knock-in mouse of maybe potentially improving executive function and then also potentially improving mobility. And kind of a conceptual model that I have for this group is that, and this is for balance, but it works for walking as well, is that, these older adults who get cancer and chemotherapy, and cancer in chemotherapy, while it's many mechanisms by which they can injure many different organ systems, the best overlying hypothesis is a mechanism of advanced aging and accelerated aging. Either way, cancer and chemotherapy, impair intrinsic capacities that are needed for balance and gait, specifically integrating sensory information. So, vision is important to consider, vestibular system important to consider. This is a nerve, and so that chemotherapy-induced neuropathy, muscle function, sarcopenia, myosteidosis, and then cachexia, all related with cancer. And so all these components that are providing information, sensory information for you to keep easy automatic control of your balance, may be impaired. And we know that some are impaired. And what your brain has to do when you're balancing is to integrate those sensory stimuli, reweight those sensory stimuli, or maybe even inhibit some of those sensory stimuli. And so in this way, central control using your brain, and specifically that dorsolateral prefrontal cortex that's known for sensory integration, is important, and maybe even more important in older cancer survivors compared to their cancer-free peers. And so maybe this targeting of mobility with neuromodulation intervention could be really successful with this population. And with that's what I got.
Thank you so much. And I think thanks for drawing attention to this really important issue. I think thanks to everyone for all the effort you put into your slides and presentation. I think it was great to see the different kind of unique angles that you bring to the attention of our viewers and to each other. So with that thought, I was curious if each of you, might have questions for each other, and we can maybe start the Q&A there.
I could jump in. Brendan's my KO1 is being reviewed next week. Yes, that's when study section is financial BI. So that's not quite nervous yet, but yeah, I know how you feel. Cool that you submitted that. I just had a question, just thinking about the PFC activity in this, in terms of your neuromechanisms, chemotherapy impairment, going back to even stuff that Mark talked about with reduced activation or higher activation. This is kind of a topic of, are we talking about greater neuroefficiency or less neuroefficiency? I think it comes down to whether you have the same performance with greater activation or lower activation. But anyway, what PFC activity pattern do you expect to see from a positive effect of the TDCS?
That's interesting you asked that, because I was going to ask that to the group in general, is after TDCS, do we expect there to be less activation, less FNIRS or more FNIRS? And so, I'm glad. I think the FNIRS is the hardest thing to, I think, defend as I talk about my aims to people. It always has been. But what we're thinking about is the increased neural efficiency. And so we're hoping that the FNIRS activity actually declines. But as you say, it's all on the matter of fact of what's happening with performance, right? And so for my thing, I think it's complicated because we're doing a dual task protocol. So there's two aspects of performance. One is the cognitive function task, like, are they doing A, C, E, you know what I mean? Are they getting those right? Is that speed, staying the same, correct speed? And how is their mobility looking? And how is the gait speed, the variability, and the symmetry performing on those things? And so I think, I'm not sure if people know the work of Andrea Rosso, but she has developed a metric to try to put all of the, specifically with dual task walking, trying to integrate those three components, being the cognitive function performance, the FNIRS performance, and then the dual task walking speed. And so, yeah, no, I get worried in the literature because I see some things with that crunch hypothesis type of deal. If all of a sudden you don't activate, or if you're activating more neurons, are you also performing better? Or or are you activating less and performing the same? Like, both those could be good. At least in my very simple understanding of FNIRS, but one of you Marks might have a better thoughts to that.
Well, Mark, you may share your thoughts on this. Yeah.
Yes, we've looked into using NIRS. We did a study with dual task walking, and this idea of efficiency is the easiest way to try to comprehend the complexity of what the NIRS is measuring, and it's a simple way to understand it. So the other aspects from a practical point of view of experimental design and the set and the protocol. So how do you have rest periods that you're comparing the dual task? Are they standing when you do this? So they're going to be quite important when you're looking at posture and when you do the assessments to compare a baseline, because everything is a change. And also, what is the system that you're using? The NIRS system that you're using, is it a...
I'm not, I think it's an Artemis. Artemis is the FNIRS system. Yeah. And I think Ted Hupper is kind of one of the people that's helping to do the analysis for that. I think, yeah. And so I'm lucky to be at Pitt for that type of stuff. And the way we're doing the stimulation is, they're not doing any active tasks during the stimulation. They're just seated for the stimulation. And then we would, do our stand, like they'd sit for the 20 minutes of stimulation. It's not stimulation during the dual task walking. Okay. And I'm kind of taking this protocol kind of from a protocol that was very effective with Dr. Brad Manner at Hebrew Senior Life and at Harvard. But yeah, I know, and I'm always interested too about how stimulation, during the task, and then also reading FNIRS, how that would change things and change that hypothesis potentially. So because then I would think that FNIRS would be maybe higher or something. Yeah,
during the stimulation, but if you're comparing the dual task to the single task, that should even out because it should be a similar response. We get quite consistent responses to TDCS, like an increase in the hemodynamic response with oxyhemoglobin, particularly during stimulation. So and everything on top of that can vary, particularly that our first studies, there was a large increase in the hemodynamic response, but then the differences in the task-related activity were within that. So we can see a reduced or a large reactivation over the baseline shift. So for me, having the task during stimulation is quite important for the sake of you're getting that neural circuitry. Probably all went through all this before, but activating the circuitry that's already involved. And
what fires together, wires together. Yeah.
And those thresholds are much higher for the brain to actually get over. And where just the stimulation is piggybacking on that. And the variability in the TDCS responses can be seen in the past because of not including, doing an offline stimulation. So that could be one aspect of an online stimulation. But to use NIRS to have a good baseline to compare to is quite important. So I'm sure that you've been advised already of having the same position and the task and non-task period, and then doing the task so that you can compare to.
Yeah.
And what is the montage you're using for the TDCS?
Is this going to be over the dorsolateral prefrontal cortex?
But just an anode, large pad?
We're going to use the neuroelectrics. I don't know. I'm not exactly sure what the exact montage is. Yeah. No, but I know we're stimulating the dorsolateral prefrontal cortex. It's going to be a left dorsolateral prefrontal cortex for 20 minutes. And then we're going to use some stim weaver algorithm to kind of improve the localization.
Okay. Yeah, I mean, using
so
high
people
definition.
Sorry, don't mean to interrupt, but people that might not know what the stim weaver is. So if you come in with a particular, study protocol and you're interested in, stimulating a particular area, we can sort of make it sort of, more precise to your desired effect. And so we basically have a brain modeling team that will meet with the scientist and come up with a plan to really personalize that particular montage. And they can go a step further as well by having an MRI, because we get that spatial, definition that we don't have, with in the absence of an MRI.
And maybe, Brendan, also, what level do you know what amperage you're going to be using?
Wow, you're drawing me back to that proposal. Yeah, no, I think I can't remember if it was 2 milliamps or 4 milliamps, but it was one of those.
It's pretty standard.
Yeah.
Yeah. Now that literature is indicating you need a higher stimulation to really get the currents through to the cortex that you're interested in. So, and it's been shown to be as safe. I mean, when we went from large pads, that was kind of one of the aspects of the work I did, was how do you get NIRS around a large pad to do online assessments? And it came from Professor John Rothwell in London, who suggested, why don't you just use high definition? So that got me to place those electrodes in that high definition format. In itself was better to localize, and it also allowed the NIRS to go around it. And of course, EEG. And, what we notice is that to reduce variability and to get more current for people that have non-responders could be simply just increasing the current. I think that's a direction that the field is going towards, is going to 3 and even 4 milliamps to really get everybody past the threshold and have the polarity changes and entrainment changes occurring.
Yeah, I just looked at our protocol. It looks like it's 2 right now, but that's good to know. Yeah, that'd be an easy.
And the safety aspects have been published now. So for 3 and 4 in older populations, so people can start to use those higher currents.
And I do want to draw attention to Mark Hartman's the statistics on people reporting that they don't have time to exercise, and then looking at the statistics on how much we're doing screen time or just kind of watching TV. And what's kind of interesting is I'm thinking about, like, let's say people are suffering from depression or anxiety, and when they go to their primary care physician, a lot of times, just from a simple, survey, they'll get prescribed an antidepressant without asking the person, are you sleeping well? Are you eating well? Are you exercising? Because I feel like we've sort of gotten away from the basics. And when you think about, our human nature, right? And as much as we all love technology, it's not really healthy to be in front of a computer, a technology all day without, breaking it up with some form of exercise, or like, our true nature is within nature, right? So what type of an impact is that having on mental health? And I don't know that people are discussing this or thinking about it, because I don't really think it's inherently in our nature, and to kind of to process all of this, like what type of effect is that having on the brain? So it's just, I hope there are some primary care physicians at some point watching this, because I think it's stunning to see those statistics. And yet we don't have time to take an hour to go for a run or go for a bike ride, or, and I think it's so crucially important to our well-being kind of overall. So
Actually, JT, that's interesting. I mean, when you brought up these questions earlier, and kind of when I proposed the last slide on mind sense, was developed for this purpose of knowledge workers, which we all are. We're in front of a computer all day and taking these breaks. I mean, of course, going outside is ideal, but when you can't,
right,
having a breath, 3-minute breath work session. And although you're using the technology, in that case, just doing a slow breathing, paced breathing, where you're synchronizing to the sound, actually detracts you from the work that you're doing, and you can't focus on anything else. So you get that rest. So this is the work I've been looking at, is to how to enhance the rest. And really, if it is rest, like a deep sleep rest, can you induce that? Can you get to that level? And a breathing task or meditation or hypnosis can get you back, not as well as probably deep sleep or having going home and having sleep, but while you're working during the day, at least for for people who cannot escape that, taking breaks and doing these kind of exercises have been shown to enhance parasympathetic response, so the autonomic nervous system, which seems to be dysfunctional in a lot of anxiety cases, and possibly even depression. So if you can, with the slow breathing, for example, enhance the parasympathetic tone, you could physiologically push yourself out of those states.
Right. Yeah, no, amazing. And you said that's going to be available shortly. You said maybe a month.
Yeah. Well, in the next few months, we're just finishing off, and it's a simple app that you could quickly just do in an office setting. And of course, there are other applications. Pardon?
Sorry to interrupt. Can you wear this during exercise? Is there any
I mean,
sweat or anything like that? That would
because it's the system is developed for this purpose of exercise itself. So we've been able to modify it for the brain, and it's ideal for that situation. Of course,
excellent.
You'd know all the issues of movement and artifacts, but in this case, it's fitted nicely, and it's very quick setup.
Well, JT, going back to what you said in terms of the screen time stuff, I mean, I think technology can be used in a way where you can have your screen and exercise. I mean, all properties that have been embedded in our smartphones just need to be, directed towards good. I think Duolingo has done that. It took all the properties of like Candy Crush and all these games and made it into an educational platform. And we just need someone to do that with exercise. I mean, I don't know if you remember Pokemon Go. All the youth in the world got very active for a short period of time, playing that game, running around the city catching Pokemon. And so I think there's just an aspect of how can we use this technology to increase physical activity by gamifying it to some degree. And for someone like me, I would love to gamify it by looking at my brain while I'm running. That would be motivating to me. That'd be cool. So
that would be amazing.
There's avenues that technology can be enhancing.
Yeah, and I think an avenue to that. Sorry, just is like that VR system that Muthalib was talking about. And so maybe we could wear a VR actually while we're exercising and look what's happening to our brain. I think that would be amazing. So Brendan, go ahead.
No, I think JT, you brought up an amazing point that I think that highlights, I think, a very clinically potential impacting part of Mark Hartman's research is that I'll say for once, from a clinical aspect, I think a lot of clinicians are asking about exercise and diet and stuff like that, but patients simply don't do it, and it's a lack of motivation, and I think that the thing is that if you could improve their motivation, tolerance to discomfort, ability to maybe handle that uncomfortableness, you know what I mean? And and if that, and what like, the way I could see it intervening is like, oh, you're not doing your the 150 minutes a week. What is the barrier for that? Oh, you feel like, you can't do it because it's too uncomfortable. Could you do a TDCS protocol to integrate, a new exercise program? And I think, out of anything you could prescribe in medicine, exercise is the best thing. You know what I mean? Like, I always call it, whenever I see a randomized controlled trial with exercise, it's like cheating. You know what I mean? It improves everything. You measure sleep, you measure cognitive function, depression, anxiety, physical function. It's like, of course it worked. If it didn't work, like, why are you publishing that it works? You should publish if it doesn't work. That'd be more novel. You know what I mean? To a certain extent. But yeah. So I feel like, I feel like you could get that cheating aspect, if you could really, impede someone's or if you could stop people from not wanting to exercise, that would be great, or make it easier. I think that's huge.
Right. So that's why I think, Mark, that a lot of the work you're doing, it was so phenomenal, because I we get that a lot when, people do ask, well, exercise is part of your treatment plan, but they just like, I can't get motivated. But like, maybe so Mark, would this be like something that would need maintenance? I don't know if like how in depth, like how long-term the study was, but is this something that, something kind of clicks on after a certain amount of sessions, that people actually, it changes their perspective on exercise, or is it something that they would need maintenance? It probably I'm sure there's like variability, but have you seen that? Or
Sure. Well, like a single session is can increase how they, the feeling on the feeling, but in terms of more of a long-term maintenance, which hasn't really been done, I think it does require multi-session. If you look at the cognitive neuroscience literature, which has done thousands of studies about executive function, working memory, multi-session, working memory can improve, but it's sustained over a 6-month, 12-month follow-up after 10 weeks of 20 minutes per day, for example. So the long-term stuff is going to have that lasting effect on adherence. Ideally, we don't want to be having everyone stimulate their brain forever. Although I guess we could, people take antidepressants forever. But the stimulation seems to be most effective for an 8 to 12 week period, after which the neuroplasticity has occurred and it's sufficient enough to sustain that change over time. Again, we've seen this moderate to large effects in the cognitive neuroscience literature, especially in working memory. So I think that the same would apply here for the exercise.
Yeah, that's amazing.
And if you got someone to and I feel like the best, another great brain stimulation is exercise. You know what I mean? And so if you got them to actually start doing the exercise, potentially after that 6 months, and you got them to sustain the exercise, and now the exercise routine is kind of integrated within their routine, maybe you don't need it then.
And the fitness.
Yeah, exactly. So
And I'm just really curious. I mean, I was not aware. I mean, I think we all have heard that, chemotherapy has side effects, but I think just like the focus on the balance and, movement, I think it's great to draw attention to that. How much chemotherapy does a person need? And I'm sure there's variability, but like to see that effect.
Yeah, it's all very variable with so we like to say chemotherapy induced peripheral neuropathy, but really every type of chemotherapy is not neurotoxic, and every type of chemotherapy has its unique mechanisms and how it does it. But 30 to 70% of people get impairments from chemotherapy in their nervous system, either peripherally or centrally. And I think the cognitive aspect is really understudied. It's for many different reasons. They don't see frank impairment on neuropsychological testing, and also neuropsychological testing to do it. People don't get in and for like 6 months later from a clinical aspect.
Right.
So it's really hard to get these assessments, and they're not 2.5 standard deviations different, but on patient-reported measures, they're talking about really frank impairment, and a lot of that impairment is things like, oh, I walked into the room and I forgot what I was doing, like, or like I was, it's really hard for me to go back to work and handle 2 tasks at the same time. So it's like a lot of these dual tasking, significant higher levels of attention and higher levels of executive function that sometimes aren't always routinely assessed. And I think one thing that was really interesting is there was a study that came out this past year that breast cancer survivors that were over the age of 60 and received chemotherapy, they're at higher risk of dementia, and vascular dementia, and and frontotemporal dementia.
Wow.
I think that that's an interesting thing to think about, in addition to depression, anxiety, and everything else
there's been, I mean, in recent years, there's been a lot of research too on showing the relationship between obesity and developing cancers and different types of cancers. So, this seems that if we can get people to, to motivate them, through stimulation and get sort of that brain plasticity going till they do it on their own, that we could, potentially like prevent more incidents of cancer, which would be beautiful. But
the other thing that I see, the other thing I see stimulation with in the cancer survivor population, every time I treat it, like I'm involved in the care of a cancer survivor, what they don't want is another medication. Like any type of another medication is just like, they're like, no, I don't want another medication.
Right.
So
exactly. Yep.
Yes.
Yeah. So I think the more we can step away from that, because unfortunately, with every medication, there are side effects. So, if we can do something that is going to kind of, minimize side effects and enhance, well-being, I think we want to get the word out. So, I mean, for the two of you that are, waiting to hear back on the KO1s, best of luck. And and I'm sure like it'll go according to plan. So I wouldn't worry. Are there anything else that I know that we are pretty much I think so far this has been the most timely discussion panel. So thank you all for really paying attention to the time. And I feel like, there's so much more that we could talk about, but I want to be respectful of everyone here. I know you've already spent an hour with us. So thank you so much. Are there any kind of last thoughts that you would like to see, either with, researchers or in the society, that a message that you want to send out to the viewers before we wrap things up for the discussion?
Maybe I can hop in there. What I see is that this exercise, it's rekindled my interest. I mean, I came from exercise physiology background, and to see this work that you guys are doing is phenomenal, and to show how the stimulation can be applied for these, enhancing motivation and for patient population is great. So, and goes down to the point: how do you get this out of the clinic? Get them to do this at home. That's where they're spending most of their time. So this aspect of developing, going back to technology itself, apps that they can, you can interact with your patients, and, will be quite important how to develop those. We're trying this direction with MindSense is for this purpose, is to have this interaction with the client, with the patient and the clinician. And you get the physiology. So I know Neuroelectrics also have a home-based system that you can monitor your patients at home. So I think this is the next frontier, is getting all this research at home and building the infrastructure for it. So
anyway, well, I just want to mention, as both of you are thinking, just to point out that I think anytime you get a diagnosis, cancer, neurodegenerative disorder, COPD, I mean, just getting the diagnosis, I feel, is traumatic, and so I think that sometimes does not get recognized, and that, people should sort of, reach out and, talk to someone if they can, that, it's, or even having trouble walking or with balance. That's traumatic. And especially, for a person that is very active, imagine, a person that, might run 100-mile marathon, right? And then they, years down the road, get a diagnosis of cancer and have this chemotherapy. And now they're having problems with, balance and gait. And that's just very traumatic. So I think it's just important to kind of recognize what, people in that situation, what they're going through.
I
Wrap
just want
things
to
up.
thank Mark, Brendan, and JT, and Neuroelectrics for putting this on. It's really cool to see other research, especially using some of the same equipment, and just see the diversity of uses, and also just to focus on the brain. It's really cool to see just more and more brain research, which has really developed in the past 15, 20 years. And even just things like cancer survivorship that I never really thought about. That's really cool to see. And just some of the discussion we've had here today, the MindSense products, and just the idea that we should, being able to get some of these technologies into the home, because they're showing more and more to be effective, is really cool and promising. So I'm excited for that.
Thank you, Mark.
Yeah, I'll just share the same type of optimism that Mark said. I've written down papers from both of the Marks that I think I'm going to go back to and to read. And, as someone who's just entering this field, I'm very excited.
Excellent. Well, thank you, everyone, all the speakers, all the viewers. Thanks for joining us today. Feel free to reach out, info@Neuroelectrics with questions. And I think all the speakers shared their contact information. You can reach out to us. We can share it with you if you don't have it. And we'll see you hopefully this fall for suicide prevention through adapting brain circuitry. Thanks, everyone.